Hey guys, so we're going to continue our discussion on biology by going over reproduction, and this is definitely some high yield content, so let's lock in. So we need to go over the cell cycle and mitosis, right? We need to know interphase is 90% of the cell cycle, and interphase is made up of the G1, G2, and S phase. Okay, now what happens in each of these phases? So G1 is where your cells grow, and this is a checkpoint, right? You want to make sure your cells have grown enough. If you pass the G1 checkpoint, then you move into the S phase. This is where your chromatids form and we have more DNA replication. After the S phase, then we have G2 phase. This is another checkpoint to make sure the cell is ready for mitosis. And so what is mitosis, right? So mitosis is split up into four: prophase, which is where your chromatids condense and your spindles form; metaphase, which is where they line up in the middle, so your metaphase plate is in the middle; anaphase is where your sister chromatids get pulled apart; and then telophase is where your nuclear envelopes reform and then they split. Okay. For chapter 2.2, we're going to talk about meiosis. So this is a little different. It's pretty much the same thing, right? So there, in this time though, you produce four non-identical. So it's like you're going through mitosis twice. So mitosis 1 will celebrate homologous chromosomes. Mitosis 2 will celebrate sister chromatids. And then you need to know, this is very important, you must know that crossing over happens in prophase 1. And then you need to know that crossing over is the reason why we have genetic diversity. Okay, and then independent assortment happens in metaphase 1. Okay, so this is very, very simple stuff. Good foundation. We're going to build a lot more on this as we go on. So I'll see you guys in the reproductive system.